Hello and welcome. I'm Victor San Pedro, and I'll be your MC for today's webinar, Proteomics in Focus, Leveraging LCMS Technology for Precision Research and Diagnostics, hosted by the Protein Proteomics and Metabolomics Facility, the PPMF, of the Philippine Genome Center, along with Medical Test Systems Incorporated and Thermo Fisher.
Good morning, everyone. We at Medical Test Systems are very proud to support this event by the Philippine Genome Center's Protein, Proteomics, and Metabolomics facility, and also to support their pioneering research. It's inspiring to see Filipino scientists attending, and we hope you will be able to use this knowledge on LCMS proteomics and the proteomics workflow for your own studies. By providing reliable, high-performance instruments and reagents through Thermo Fisher Scientific, we hope to continue to enable cutting-edge research that advances science and strengthens the local scientific community. Thank you, and please enjoy today's webinar.
All right. Thank you, Jessica. Today, we dive into the transformative world of proteomics, where LCMS workflows are reshaping diagnostics, agriculture, and biomedical research. You'll hear from three distinguished experts who will share practical innovations, breakthrough methods, and real-world applications in precision science. After all the presentations, we'll open the floor for a live question and answer. Please stay until the end to complete our evaluation form and receive your certificate of participation. Licensed chemists and chemical technicians will also earn 2 CPD units for chemistry. Let's get started, shall we? By the end of today's webinar, we hope you'll leave with a deeper understanding of cutting-edge proteomics technologies and how these tools are being harnessed to advance diagnostics, therapeutic development, and systems biology. Now, before we begin, a quick reminder, please hold on to your questions until the end of all the presentations. So our first speaker is Dr. Neil Andrew Basto. Yeah, okay. Dr. Neil is a molecular biologist with deep expertise in protein science and multi-omics research. He earned his degrees from the UP Diliman and Tulane University and currently serves as an associate professor at the National Institute of Molecular Biology and Biotechnology, principal investigator of the Protein Structure and Immunology Laboratory, and program director of the PPMF at the Philippine Genome Center. His research spans Sardinella proteomics, integrine structure, SARS-CoV-2 plasmid proteomics, and many more. Let's all give a warm welcome to Dr. Neil Andrew Basto.
Good morning, everyone, and thank you again for joining us for this proteomics webinar. Again, what we're hoping to do today is to present different ways in which we can do actually multi-omics analysis, but focusing on the proteomic side, and then looking at current applications that we would have for this, as well as some advancements that are actually there in terms of the technology that makes doing these things a little bit easier. So, of course, we thank all our co-presenters for this event, and we do hope that you consider their different products, and applications as we try to all do proteomics here in the Philippines. Right. So let me start our presentation. Wait a minute. Sorry, we're just having some slight technical difficulties. All right. I just like to confirm with the team. Can we see this? Can we see the screen? Okay, that's good. So again, I'd like to welcome everyone to this proteomics webinar. And I would want to focus in this particular part of the session on my talk with one of the things that we most deal with when we try to work with proteomics, and that is biomarker discovery and its eventual applications. Now, but before we delve in to proteomics itself as a focus, I think we really should look at the omics technologies to begin. No? And then, of course, when people think about omics technologies, one of the most common topics that they would think about would be precision medicine. And what is precision medicine? Well, simply speaking, this is providing the proper medication for a patient based on their determined sensitivities for treatments. In Filipino, this actually quite elegantly stated as akmang gamot, no? And akmang gamot doesn't just mean the medication. Sometimes it also means giving it at the proper time in the proper way. No? So it's akmang gamot sa tamang panahon. No? And to achieve this goal, we really aim to achieve better detection techniques, better diagnosis, and that eventually translates to better treatment. Unfortunately, much of our common medical treatments still rely on what we call trial and error methods. Often, this means that we try a general treatment, and then when that doesn't work, we try a different method. Now, such methods are actually quite costly at best, and in some cases may even prove to be fatal. With lives on the line, physicians never take the decisions on treatment very lightly. No? We should never make them blindly and without sound basis. And that really is where biomarkers come in. So biomarkers, as their names imply, mark or denote existence or occurrence of a specific condition. In the case of diseases, these may be markers of a genetic predisposition or markers or symptoms of the diseased state. Throughout the years, several biomarkers have gained predominance. For cancer, for example, I think we are all familiar with how P53 is a major mover. No? And back when I was in grad school, it was actually the time when the BRCA mutations were first coming to light. No? And that's actually grown in terms of the knowledge that we've had in terms of their relevance. And in fact, more recently, its significance has been actually been put into the limelight. No, its significance for providing risk for breast cancer has been put in the limelight by several celebrities, no, that have tried to espouse why testing for these mutations are actually quite important. Now, some established biomarkers indicate not just the predisposition for a disease, but the actual dysfunctions that may be happening. Now, in the case of diabetes, monitoring your fasting blood sugar and HbA1c levels can actually indicate if your body can still properly metabolize what you've been eating. No? And in the case of HbA1c, how often you've actually indulged in cheat days in the past few months. Right? So, meron tayong ways in which we might be able to see if an actual dysfunction is actually happening. And in some cases, we actually might be able to see how often that dysfunction has been pervading, depending on what we try to monitor. So it depends on the type of biomarker, what we are able to discern, no? What type of information we might be able to discern. Often, no, the biomarkers that we have, no, while they would be good signifiers of a particular dysfunction, sometimes, no, it's not quite enough, no, in terms of being able to describe what is actually happening within a system. This is particularly true when the biomarkers are being applied to things like complex diseases. So unlike, for example, the poster child for single mutation analysis, which is cystic fibrosis, wherein a single mutation can cause the defect in the protein. Sometimes it's actually multiple mutations that are happening that are part of a particular dysfunction. So in these types of complex diseases like cancer and diabetes, often the biomarkers and therefore the factors that play a role in the onset of the disease are actually multiple, or they're called multifactorial. In fact, in terms of the BRCA mutations, no, while it's known to be a known biomarker for cancer and breast cancer in particular, or ovarian cancer in particular, you can note here that in the standard cancer panels that people now do to look at genetic predisposition, they're only one small part of the many, right? So we see here that sometimes we have many types of biomarkers that have to be looked at in order to get a better idea of what's happening. In terms of diabetes itself, there are also many genetic predisposition markers that have been identified, right? And this would come in the different forms of diabetes that are present. And as well as the functional biomarkers. No? So there are several functional biomarkers. There's creatinine levels, there's cystatin C, and these have all been related to the disease. Now, the multifactorial nature of diabetes and its progression is quite colorfully represented here. Right? It's showing you the multiple bases of staging diabetic nephropathy and its progression. And these actually need to be verified, not just in a single time point, but often, no, if you want to get a really good diagnosis of how true the observed nephropathic stage is existing, this type of test actually needs to be done in succession over a course of about 3 months. No? So it's actually a very intricate set of tests that we need to do to be able to confirm whether or not a proper staging of diabetes is provided, and therefore the actual treatment that is to be given will be prescribed. So despite the complexity, the key to providing precision medicine, to all types of diseases really comes down to understanding the disease and its underlying systems. And therefore, when we try to do this, we must acknowledge that there is a need to look at the bigger picture. And that's actually what we achieve when we do multi-omics analysis. So multi-omics analysis provides us that big picture, right? It allows us to look at the many aspects that may come and provide the state of the disease. Now, and then this could involve looking at whether or not there were genetic predispositions that were already in place, and then adding on to those genetic predispositions the actual translation of them into what is, let's say, expressed into the functional proteins that are there and the corresponding metabolites that they provide. That gives us a bigger sense of what is happening in this particular individual, and therefore, if it is a disease, how we may best treat that particular disease. All right, so in our laboratory, now we aim to help provide multi-omics analysis, right? We are part of the Philippine Genome Center. The Philippine Genome Center has made its name in its capacity to do a lot of the genomic and transcriptomic analysis done in the country. And the PPMF is tasked with providing the proteomic and the multi-omic part of the picture that we can provide for multi-omic analysis. And we are able to do this because we are have at our facility the means to utilize the highest resolution mass spectrometry system in the country. So we have a Thermo Fisher Orbitrap Fusion Tribrid, and we are able to analyze in a sample the complete sets of proteins and metabolites that they have. And these are what we call their proteomes and metabolomes. And we therefore are able to look at these different samples in different conditions, say healthy or diseased. And by identifying each individual protein type, we can therefore try to understand what's actually happening in each of these samples. So if we're, let's say, trying to look at a sample that is healthy versus that of a diseased, what is different between the two, and why exactly are we seeing the symptoms that are observable in your diseased individual? So how do we do this? Well, again, let's take a closer look at this high-resolution mass spectrometer. It is based on mass spectrometric technologies, which means that we are able to look at components within a sample, essentially by differences in mass of, well, the different fragments that we generate from them. So let's take a closer look at our Orbitrap system, look at its component parts and try to see what's happening here. So what we're showing you are important sections of the Orbitrap system that we have. On the top right, we have, well, it's demonstrating how after passing through a selection column, the different fragments that are taken from a sample, these are previously cut up into their component fragments, are ionized through an ESI system. These are then selected for through a quadrupole system that is passed through, that's shown on the bottom right. And then afterwards, depending on the types of ions that we would want to analyze, we can select for them and eventually characterize them using our Orbitrap mass detector. That's the one on the bottom left corner, right? So as the peptides are ionized, sorted, and selected, and eventually measured, we determine their mass-to-charge character. And from these observed mass-to-charge ratio values, or MZ values, we can determine the identities of the component peptides and subsequently the proteins from which they are derived. So just to show you what happens there. So this is a typical MS spectrum that we get. And for each of these peaks, we are able to determine the types of fragments, mass fragments that are there. And so we see something like this, right? Where the graph shows you observed mass fragments. And depending on what we expect, what we expect in terms of the types of sequences, let's say, that these fragments can come from, now we might be able to identify which fragment corresponds to whatever sequence. So what we're showing you right now on the screen is a quite simplified system for that. Like it's just showing you the B and Y fragments. But consider that we have databases that have the corresponding masses for different peptide forms that may be found in different peptides. And therefore, we then try to match the observed fragment types onto these databases to determine which of the proteotypic peptides we might be able to identify, and therefore, correspondingly, be able to determine if a particular identified protein is present. So the higher number of the expected fragments are observed and are identified for a given protein, the higher the confidence that we have that the source sample actually contains that protein. And of course, we utilize a lot of quality control and statistical measurements to confirm whether or not our assignments are correct. And once they pass through these very stringent QC control, we have with enough confidence be able to say that these are the different peptides and these are the different proteins that our different sample has. So we therefore apply that type of analysis through for the different samples that we can analyze. So again, we can compare healthy or diseased or different stages of disease to be able to find out what is there. Okay, so after having identified the different proteins that are there, we can then now try to compare if the same proteins are found in the different samples, or in the cases of a pairwise comparison, are we able to say if it is up or down regulated in one of the samples or the different types? So this type of analysis lets us determine what we call differentially expressed proteins or DEPs. And in this paired analysis, we are actually able to show which proteins are up or down regulated in a sample compared to a given control. So and again, this is a paired analysis. So this usually means that we try to essentially compare two conditions, and it could be as simple as a binary diseased versus healthy, or it could be looking at different stage severities. Okay, so I would want to emphasize, though, that when we want to do these comparisons, and for these comparisons to be quite reliable, a very sound research design has to have been employed, meaning when we do the sampling in the defined study groups, we actually have to be quite clear that there should be a sound basis of how they're separated into the different groups, so that when we do make comparisons between the different groups, it will be more likely that we find distinct differences between them. So, of course, we need to be able to verify that, assuming we put in as much effort as we can into the study design and hope that the different study group members are as they were defined in the onset of the study. But we still need to verify, are they actually distinct? Are they behaving differently? And so that's where doing multifactorial analysis will actually do. So we do principal component analysis or PCA plots. And then this multivariate analysis, we might be able to say if two groups, or if the samples that we studied, may they be in different groups or similar, do they cluster into different groups? Now, are we able to distinguish them from each other? You can see here in this sample data set that sometimes that's not quite the case. I mean, you could see a little bit of clustering happening here, but you can see that at least in this data set, based on these particular parameters, the distinction between the two groups isn't quite distinct, right? Sometimes we're lucky though. Sometimes we're lucky that the actual factors that we're considering for the PCA analysis actually does provide a good set of clusters that are quite easy to distinguish from each other. And therefore, the comparisons between them would be much easier to do. No? So here's showing you a PCA plot where the samples that we see do cluster into very distinct groups, which then can then be compared. Okay. All right. So if whether or not it is of this case or another case, if we want to do a comparison to see which genes are up or down regulated between paired groups, we again do a volcano plot, which is what we see here, right? And therefore we see what are differentially expressed. Again, we show on the right, on the colored red, those that are higher in expression compared to the control group. And then blue on the left shows us the those proteins that are of lower expression than the control group. No? We also see here, no, that sometimes it's just it's not just, no, it's not just the fact that we know which proteins these are, right? That's important. So of course that's an important piece of information. We want to know what is up or down regulated between the conditions. But taking that a little deeper, again, with the concept of omics making or looking at a bigger picture, right? We don't want just to look at the identities, but from the identities of those that are defined to be differentially expressed, we might want to see how they are related. And that's where the pathways, no, the cellular pathways in which these different differentially expressed proteins might belong would come into importance. Meaning, if I know that proteins X, Y, and Z are upregulated, how does that help in the observed symptom? And we might want to, or we might be able to see that by noting that all proteins X, Y, and Z actually belong to the same pathway, right? And I also want to mention that it's not just the upregulated, right? Sometimes it actually has to deal with proteins that are also downregulated that might actually affect a certain cellular pathway because they are downregulated. So to reiterate, the volcano plots give us identities of what is up or downregulated. And to take that further, we look at how the different components that are found in there are actually part of different pathways, right? In this case, for example, we show you that there are certain key cellular pathways that are enriched, or meaning there are high amounts of differentially expressed proteins that are found in these pathways. In this case, platelet degranulation, perhaps, or complement activation in this particular case. No? So we are able to do that and therefore try to find different pathways that are affected in a given condition. Now, perhaps it is actually easier to recognize how the individual components that are differentially expressed affect pathways by looking at the pathways themselves. So here's another representation of how that may be. So we might look at the full cellular pathway that is present in a given system, right? And be able to identify from there, what components are affected or are observed to be affected in the samples that we study. So we're showing here an example of a represented cellular pathway, one for cancer, right? And if we look deeper, or look a little closer onto that representation of the pathway, we are able to see the different proteins that are known to be part of this pathway. And some of them are actually promoters, meaning their function leads to the further progression of a certain function. Or alternatively, there are some that are actually what we call inhibitors. So the function of a pathway depends on controls, modulation controls, some that need to push the progression and some that need to keep it in check in order to have the proper balance and it to function properly. Okay, so that's what we do. And then often these different proteins that we have observed to be differentially expressed are actually what sometimes serve as biomarkers, meaning if they're highly expressed, they can actually serve as a biomarker for diagnosis, or in some cases that we really can place them as part of the pathway, then we now have targets for modulation and perhaps possible treatment opportunities, for this particular type of disease, right? And so to recap, no, that looking at the pathway and how the different components work is a representation again of how multi-omics analysis, which gives us the capacity to look at that bigger picture, actually gives us ways in which we might be able to find different applications that could be relevant to the health industry, in this case for diseases. And similar processes would, of course, be important, let's say, if we want to look at better production, no, of certain things for industry, or in some cases as well, we might want to look at what are more easily identifiable, in cases that might help the forensic applications. Okay, so there's many applications that we have. I'd just like to share with you a few of the projects that we have here in the PGC PPMF. So we have projects that deal with SARS-CoV-2. This is a proteomics research project that we have looking at total protein character of different stages of SARS-CoV-2. And then we have a metabolomic study on the progression of diabetic nephropathy for T2DM patients and healthy controls. We also have a proteomic analysis to look at the relevance of particular biomarkers as observed through ELISA and HRMS, right? And also, we're starting another study on diabetic foot ulcers. Now, this is partnering with De La Salle University and actually many other institutions. Right. And so there's a lot of applications that are being done, and I think there's definitely a lot more to be explored. The past projects have been, being actively implemented really with a lot of healthcare partnerships that we have with many different institutions, all working towards the goal of better precision medicine, better diagnosis, better treatment. No. This set of research has provided a lot of new information. We will be publishing those soon, but they will be focusing, of course, a lot of that, what is expected of us there would be to find, as we had shown the importance of the different biomarkers that may be important for, let's say, diagnosis or treatment or of the different diseases. No? So there's a lot of work that has been done. We would want to be able to work more on these topics, and we would definitely want to work with you because PGC's goal has really been to expand omics capabilities here in the Philippines, and I think capacity enhancement really just involves more than acquiring technology. We need the cooperation of the many stakeholders that will ensure that this technology will actually be put to good use. So we are the Philippine Genome Center, and we hope you can help us expand omics capabilities for a better Philippines. Maraming salamat po, and I hope everyone has a good day in this webinar. And I hope that we are able to help you do more proteomics research. Maraming salamat po.
Thank you, Dr. Vasquez, for discussing biomarker discovery workflows and showcasing the proteomics capabilities here in the Philippines. We hope today's talk inspire more local researchers to expand omics technologies. And just a reminder to please hold on to your questions until the end of all the presentations. So let's take a 30-minute break. Feel free to stretch, grab a drink, and enjoy our promotional segment. We'll be back at 10 a.m. And good morning and welcome back to everyone. So we will now entertain questions for Dr. Neil's presentation. So just type it in the question and answer box. Yeah.
Good morning po to those in MSU IIT. I just saw the message. So hopefully everything is okay naman po ba? Oh, I saw that there was an earthquake. Everything okay naman po dyan? So we do hope that everything is okay down in MSU IIT no po. Regarding the question, again, thank you. That's good to know. Regarding the question of the recording, we'll just have to clear it with the rest of the team because especially with our partners, no, before but we'll see what we can do regarding that, no. Sige po. If there are any questions, especially regarding the ways in which we might want to analyze, just let us know. Siguro, let me just mention that definitely the field of proteomics has gotten a lot of interest in the past few years. Yun nga, no, as we translate from a genomic level analysis, often we would want to see aside from what is predisposed, what is actually happening in a given system. And I think with proteomics, that's really where we might be able to see things, you know, because we are looking at what is functional and therefore what is providing the actual characteristics of different things. Now, while the focus of a lot of the presentation had been on health, I again want to stress that the applications of proteomics really span many, many fields. Um, in fact, I do have a lecture later on this afternoon trying to look at how proteomics and other multi-omics analysis can actually be geared towards looking at nutrition. No? So, and of course, when we look at nutrition, there's the aspect that we look at in terms of the health of the individual. Aside from that, though, we can look into what may be providing that nutrition. No? So, yung proteomic and metabolomic analysis natin, we can look at uh, the different components, the different foodstuffs that are made available to our different um, citizens here in the Philippines. So you might want to consider, for example, if your research goes into better food production, then that's one way that we can look at it. There has been much research, let's say, on trying to provide either in terms of rice. Na po? We can look at rice and how there's definitely a lot of research that's going on in the country trying to look at what factors may provide better drought tolerance or better flood tolerance. So again, a lot of the good work here has been looking at on the genomic level, but we also can look at what can be changed now given the different strains that are being developed. How exactly is it able to provide a certain strain with better drought tolerance or better flood tolerance? Partly something that we might all look into because of all the issues that we've heard regarding flood control projects, right? So there's that, you know, looking at things like rice. And then also if there are specific unique products that are available in the different regions, you know, then both proteomic and metabolomic analysis can provide a better or a more in-depth analysis of actually what is making the different products to be more unique, more special. No, so for what comes to mind, for example, would be Guimaras mangoes are very well priced, and I think it's definitely something that can be further analyzed, no, or in a way that you might be able to try to find a signature so that you can also verify, for example, if some people are claiming, you know, to be providing Guimaras mangoes, especially if they're selling it abroad or something, then maybe there would be one way to validate that depending on biomarkers that may be found on the samples. So that's one thing that can be done for mangoes, for durian, for other food products that are definitely uniquely Filipino. And then we might want to be able to really emphasize that in our own products. So yun po. So that's a starter in what we may apply for. Okay, I think
Yeah.
are there other questions?
There are questions, sir.
Okay.
So One is how can AI contribute to the study of proteomics?
Okay. So, as with most things in AI, the what is very essential would be the amount of data that's available for it to learn. No? Kasi ang AI naman, you train it on a set and you train it to recognize certain things that will mean something, right? And what that relies on would be how well populated the database is on a given topic, right? So, for example, we want to analyze the different components of a given sample. Um, we rely a lot on the databases that are available out there. AI can definitely help with that, no? Supplying it with different data and letting it search within that data for specific patterns. No, that's something that we might try to employ AI with. But I do want to note that again, it is very much dependent on the available data. And for example, that's one thing that we would want to consider, right? If our databases are very heavily based on data that was supplied by other locations, other populations, then the potential significance of a given variation might be skewed, no? To be more appropriate for the training base, for that population base. No? So that's actually why building the Philippine data is also very important. No? Um, so that we can build our own database, and then we know that the applications that we can get or the analysis that we can get from these databases would be more appropriate for the samples that they were dealing with. And that's why we're also wanting to employ uh, or to encourage a lot of people to be doing a lot of their multi-omic studies here in the Philippines, because we want to help build that database also, no? Kasi if we all do the work here, then mapapalaki natin yung database natin locally, and that means that we are also more accurate in our analysis of local samples. So I think it's something that we can all work with. Okay, thank you, Victor.
Yeah. Thank you, Doc Neil. That question was from Mr. Jesus Aquino. And next question from an anonymous attendee. Is it possible po to utilize a proteomics data analysis platform of PPMF for LCMS data obtained using a different LCMS, let's say from water or from Agilent?
Yes. Our team at the PPMF has been looking into and working with multiple platforms for analyzing data. Uh, we actually try to do that. No. So hindi lang. So there are many ways in which we can engage with the PPMF. Of course, one very parang straightforward analysis would be we do the analysis here, and then we conduct the data acquisition here, and then we do the analysis here now. And we can actually try several platforms. There are the platforms that are available through Thermo Fisher, and of course that's the most ideal in the case that we have, because it's uh, a direct analysis of the data that was acquired on a Thermo Fisher system. But realistically, the data analysis platforms can take raw data files from different mass spec types, so that we can actually compare also. No, yung what may be, let's say, the analysis that's provided to you by this platform versus that platform. So kaya naman dapat, no? But we'll have to talk with the interested researchers to ensure that the data type that is already available with them is compatible. So that's something that we can try to look into if we can talk about the project.
So I'd like to add, there's also because different platforms have different file formats for data, but there's an intermediary conversion process. But the short answer is yes.
Mm.
Okay. Next question from Mr. Montera. So, greetings. I'm a beginner in proteomics. I would like to ask how long does LCMS process usually take from sample injection to readout?
Okay. So two things. First, welcome to Proteomics. I think it's always something nice to find that there are people venturing into new fields. So welcome to the field of Proteomics. Um, on the time frame, it actually depends on the complexity of the analysis that you want done. No, if it's let's say a full scan, untargeted proteomics, strictly speaking, one injection time, one sample run for that, I think is around 3 hours. Um, so yeah, I think no, yeah, it's going to depend on the number of samples that will be done in the study, yung actual runtime that we would need to allocate for the complete analysis to be done. No, but yeah, a simple analysis could just be as short as 3 hours. So what we normally do is, if you're planning a specific study design, we would want to have a discussion with you to confirm that the planned protocol that we will employ with your project is, appropriate for the type of research question that you're going to ask. And then we can make suggestions on the protocol that we would want to employ. And after verifying that, then of course, we can provide you an actual quotation to find out how much that might need to be supported.
Okay. So we actually have another question from an anonymous attendee. So, Dr. Bascos, given the current capabilities of the Philippine Proteomics and Metabolomics facility, how do you see proteomics or metabolomics being applied in the field of forensic science? For example, in areas like toxicology, trace evidence, or biological sample analysis.
Okay. Thank you very much for that question. Well, we do have concrete plans to help our Philippine law enforcement agencies regarding this. One way that we're foreseeing that can be applied would be in the development of better forensic analysis test kits. So proteomics may be applied in that, in trying to help determine which biomarkers would be most effective in trying to identify certain sample types. No? So for example, we might be able to provide distinct biomarkers that will show you the presence of one biofluid in the presence of another biofluid, no, without question. So we're actually working here. No, this is a project that was recently approved by the DOST. It's set to be implemented starting next year. We'll be working with several agencies. St. Luke's Medical Hospital is one of them, and the PNP. So we're going to, and then the NSRI, the DNA Analysis Lab of the NSRI, we'll be working with them on this one. And what we're hoping to do is to analyze, again, different combinations of biofluids to see if within conditions that might be relevant for what they may be seen as in crime scenes, if we are able to detect specific biomarkers at certain levels of efficiency that would allow better analysis of these samples and therefore help our law enforcement agents.
Okay, thank you, sir. Next question from an anonymous attendee. So, how can data scientists be more involved in the omics research that we do?
Right. Well, first off is, I think if you would want, there's the thing with omics, right? It's a lot of data. Always a lot of data. Be it genomics, transcriptomics, metabolomics, proteomics. We're talking about the total amounts of that one type of information from a given cell. And that is often the problem, right? Meaning, it's a benefit. We see the big picture, but sometimes it's uh, so much that it's quite hard to digest. No? And I think that's where the data scientists will really be able to help, because I think, as an example, we try to look at the remember if you remember the lecture, we one starting point of the analysis would be the differential expression of proteins. No? So we depending on two different conditions, we want to see what is up or down regulated, and we have, let's say, 1,000 list of proteins that are up or down regulated. And so the individual lists are going to be quite large. And while it is possible to manually do that, right? Not to look at each individual upregulated, downregulated protein and then try to find the different pathways in which they are involved. It's the way in which you can integrate that data that I think would benefit a lot from data scientists. Meaning, we can, of course, figure out which pathways they normally belong to, but then the pathways and how they come together to the actual symptoms, that I think requires, well, one, the eye of an experienced expert, no? In, let's say, if we're talking about a health related analysis, we would want to consider the insight of the physicians, no, on how these particular pathways eventually lead to a particular symptom. And of course, also the proper handling of the data that I think will be provided to us by experts in data science. So if you're willing to do the work, or we would want to do some work related to this, let us know, and we'll find a way to work together. Thank you.
So I will take one last question from the audience. So from an anonymous attendee, to my understanding, the existing projects and collaborations are more tied with human health. Do you also have projects or plans related to animal health and diseases?
Okay. Yes, no. Yeah, as of plans, definitely yes. We and I would like to explain that the human health side is partly born of the fact that PPMF, of course, was graciously supported in its implementation by the PCHRD, which is, of course, focused a lot on health research, human health research. But we recognize the One Health policy, and definitely we look at possibilities of finding how animal welfare, animal health will eventually lead back to eventual effects on human health. So definitely there's a lot of interest there. In fact, um, I know the PGC is involved right now in some work with ASFE, and we're looking into ways in which we might be able to help contribute with that as well. And so things like that, no, I think there's definitely a lot of interest to be able to do that. We would want to help. That's not the question. And in fact, I want to emphasize that the PPMF is that type of facility that is not restricted to one field. Its main goal has been to allow multi-omics capabilities for multiple fields. And because of its setup, it is able to do so. No? So it's just a matter of being able to partner with different labs that have direct focus on that. No? And we're looking, no, where we're right now, one of the projects that is already related, we're looking at Tawilis and its proteomic analysis, compared to its marine sister species. But that analysis can be expanded to other fish, other animal types, and we would want to help if you can give us targets. So let us know and let's see.
Okay, Sir Neil, thank you. Can you take one last question?
Sure, go ahead. Thank you,
Ayan.
Victor.
Sige po. What would you say are the main limitations to be considered in a proteomics analysis from an anonymous attendee?
um, the main limitations would be the number of samples that you would need to be able to make a statistically sound answer to a particular given research question. So that's one of the main considerations because, as mentioned previously, for example, compared to metabolomics, proteomics requires much more MS runtime. So because it deals with more complex samples, it needs to be able to run at the MS system in quite much longer. So to give an example, let's say from 30 minutes to 3 hours, no? Or 15 minutes to 3 hours in difference natin, sometimes depending on the complexity of the sample. Now, that being said, I mean, honestly, it's going to cost more in terms of funds to run a proteomics work than a metabolomics work. But I think the benefit again to this would be the more direct analysis of what might be happening. No, if the research question actually deals with looking at, let's say, altered or dysfunctional protein systems within a given sample. It's very direct if we look at it on the proteomic level. So the in certain cases, definitely that is worthwhile. No? Yun yung parang despite the slightly higher cost, it because of the more direct approach, it provides us what might be a more effective answer. So, but that's something that has to be discerned at the start of the research project. So, yun yung, I think that's what I want to emphasize again. No? Always in a lot of our research projects, that initial uh, sound design, no? Initial, parang kailangan maayos yung pagkaka-setup talaga ng research projects natin at the start. Now, of course, there is leeway naman to adjust. Pero I think at the start, kailangan importante maayos yung pagkaisip natin on how these things have to be designed so that when we make proposals that will allow us to implement these projects, we are able to do so correctly. No? And for that naman, I would want to state that we are very much grateful to the government and to other funding agencies on how much support they have provided. I think the benefit of multi-omics work has really been seen, and there is much support for this field currently. And as long as there are relevant research questions that are being asked, we found that there has been a lot of support for that. So hindi ganun dapat kalaking concern yung cost because there should be ways to find funding, no, if we are able to defend the value correctly. And that is also where we also aim to help. No, we've had a chance to work with several researchers. In fact, we had a capacity building expansion program with clinician researchers, and our goal there was to involve more clinicians so that we get their perspective on what might be medically relevant questions, especially in their own locations, in their own regions. And what we did was, after doing these workshops, we worked with them to craft proposals, no? Proposals to be sent for funding. There's, yeah, parang, it's a work in progress, but definitely I think that's something that was quite successful in that we found support from different clinician researchers in actually trying to build these concrete research projects that are actually fundable. Yeah, so hopefully that helps. Yeah.
Thank you, Dr. Neil, for your questions. And now, Dr. Neil will also give another lecture.
Sige. I think we actually have just a few minutes left before Dr. Patel can make it. So, if the audience will indulge me, I figured it would be quite helpful if we went a little bit back to the basics of mass spectrometry, just so that we get a better sense of the where we get that data that we eventually translate into the proteomic characters. No, I'll just serve as the opening act muna for Dr. Patel. But it's again very basic for now. But let's see now what we are able to do before he comes in. So I think I can share. Yeah. Okay. Give me a few minutes to set up. Can you see? Okay. Sige po, no? So, again, I know a lot of you are actually quite familiar with mass spectrometry, but this very short lecture, this very basic lecture is just meant to provide some information about what went behind the development of this technique and what gave us the capacity to do multi-omics work, no, that we currently have in the facility. So, just a minute. All right. So, just to go back, no, mass spectrometry again is a technique that lets us look at what is part of a given sample. No, essentially, what we're trying to do here is we're looking at all the basic components that are present in a sample, and we are able to do that by trying to break it up. No? We break it up and then we try to find what is there. Now, all mass spectrometers, depending on while they may have had better complexities now we have as shown in the system of for the order trap, they all still rely on the fact that we break up the sample through an ionizer. We then try to accelerate the sample in some way. We charge it so that it has a charged capacity that then flies through a drift region or goes through a drift region and then eventually ends up in a detector. In the most basic forms, this would be what provides something like a time of flight measurement, right? That then tells us about the character of the different ions. Now, um, again, at the start, we have several ways in which we can try to generate these ions. And a lot of the new technologies actually try to vary on this to try to get different fragments so that we can better analyze the different components of a sample. But even at the start, they've actually found that there are actually quite robust ways in which we might be able to ionize systems. And what I'm showing you here are classic slides from John Fenn, no? And this is from his Nobel lecture where he presented the different ways in which he might be able to generate ions from a sample. This could be from electron bombardment, from chemical ionization, from the use of lasers in photoionization or in field desorption. Field desorption is what we utilize in ESI, which is what we have in the Orbitrap, where a nozzle serves as the system through which we charge different particles, and then it's sprayed through and it's ionized. In some cases, we have what we call the MALDI systems, which are the matrix-assisted laser desorption ionization technologies, wherein you embed the sample onto a matrix and then you hit it with a laser, and the explosive event that causes desorption actually ionizes the sample itself. That is work pioneered by Koichi Tanaka, and he was also awarded the Nobel Prize for this. Now, both Koichi Tanaka and John Fenn were given awards for these developments in mass spec because they actually provided ways in which they might ionize bigger fragments of samples so that in some cases, what we might want to analyze would be full proteins instead of their peptides. No? So this is just showing you how that can be done. Again, with a laser, the matrix is hit and the explosive event eventually ionizes the sample that's there. Alternatively, an ESI system or an electrospray ionization system can be employed, wherein the components are placed within droplets within a charged nozzle. And again, because of evaporation that happens within the droplets, eventually the ions are released and they are then taken into the system in their ionized form. Okay, so there's many systems that we can employ for this. The goal is to ionize samples, be it in their positive form or electric or in their negative form. Okay. After ionizing the samples, again, the charges that these things would have would affect the way in which they would travel through an electric potential field, right? And so things that would have higher charge, let's say, would probably have a greater force flinging it through. And therefore, eventually, that affects the amount of velocity that it reaches upon acceleration, right? And that may provide certain ions to be a bit faster before they get to the detector, right? So the times of flight that these things would have would be much shorter. Okay, so we are able to relate the charges and the masses of the different systems and are able to relate them to the times of flight that they reach a detector, right? I do want to point out at the bottom part of this graph how the mass to charge ratio is directly related to the time of flight. And that's why MZ values or mass to charge ratios are also a good way for us to characterize different ions, no? As they are detected in the mass spec system. Now, different samples, when ionized, usually break down into different components. And these are usually of certain signatures. So for example, this is showing an NH4 system. When ionized, can break down into 4 components, no? And that provides us like a signature pattern that we are able to see, right? Here's another example of how a very simple amino acid like glycine actually has a characteristic component pattern once ionized, and therefore we are able to recognize the presence of these components, no, based on that pattern. So here's glycine, which has an ammonia system. No? So if note that the ammonia component of glycine should give you peaks at these MZ values. And if we go back to the main structure of glycine and how it's broken down into components, those same peaks are seen here on the bottom left, near the 14, 15, 16, and 17 MZ values. So we know that glycine has an ammonium component or could have a fragment generated from it that's like ammonia because we recognize the same patterns. Other groups within glycine are also observable, like for example, your methylamine groups, no, which is this component, right? Giving you characteristic peaks at around the 28 to 32 MZ value, right? And we note that those are similar peaks again if we look at the big structure. And then lastly, your your formic acid system, right, will have the corresponding values over there. Okay, so this this process of trying to look at component parts and trying to figure out what they are making up, no, is the same process that we do when we look at peptides and trying to figure out what proteins are there. No, and what we are reliant on is the fact that many researchers have done the hard work of trying to find out the different patterns, no, that are may be associated with these different components. And because of their past contributions, we have databases, we have things like mass bank, wherein the MZ patterns in different samples are stored, and therefore we are able to denote, for example, what components within other previously archived samples match the MZ values that we get from our MS data. Okay, so this same pattern is what we apply, no, in trying to find the different proteins that are present in a different sample, and therefore we are able to designate, no, certain things to be in that sample, and therefore might serve as biomarkers for a given disease, no. Because we are able to utilize these patterns as biomarkers, again, they can be used for diagnosis. They could be for the diagnosis of a particular symptom in a disease. In some cases, if we're talking about infection, then potentially we're looking at markers that might be able to show us whether or not there is particular microbial contamination. No? Now, one might think that, parang for microbial contamination, baka naman overkill ang paggamit ng mass spec. And I guess that depends on several things, right? One thing that what I can say is to the benefit of certain mass spec systems is that in terms of identifying microbial contamination from a sample, analysis can be done in as little as about a minute. Now, if in some systems, if it's a MALDI system where you blast it and then it's able to identify some microbial specific polypeptides, no, then the determination of contamination for that particular microbe can be done in a very short time. No? And in the hospital setting, that can be very advantageous, which especially if we compare it to standard culture and sensitivity methods that might take more time, like about at least 24 hours or 48 hours. Now, so again, it depends on the setup that you have, but knowing what different technologies are able to do lets us uh, evaluate whether or not they might be the appropriate system to use in a given system. Okay, right. Okay, so in terms of protein analysis, we mentioned earlier that we are able to determine present components within a sample. Sometimes it's the full protein, depending on how it is ionized. No? So if it's a certain full protein, we might know that it exists in one form, in several ionization forms, and we will be able to denote that in the corresponding mass to charge ratios that we will get. Since it's mass overcharge, if we have multiple charges in the same target, then we can see the peak at corresponding mass to charge ratios, which should be calculable depending on that mass and charge value, right? I'm also showing here, so I'm just going through this so that, yeah, okay. This is what I said earlier regarding being able to find out, though, sometimes what we're not concerned about the main protein only, but because let's say a certain number of proteins might actually have the same molecular weight. Now, we know that to be true. But what would be able to distinguish one protein from another, even though they're of the same molecular weight, could be the way in which its sequence exists. There could be particular sequences that are available for protein A, and for protein B, the arrangement of the amino acids can vary. And therefore, if the arrangement of their amino acids varies, then if they are cut up by a protease in specific ways, right, then the fragments that they may generate could be different. So being able to decipher what the expected fragment types would be given a particular protease and the corresponding molecular weights that they should have lets us identify from the expected mass spectrum what fragments might correspond to the different peaks that are observed. So we're showing you here the way in which we try to do this. We usually call them as the B and Y fragments because let's say in this peptide, we're looking at the B fragments to be the ones generated counting the amino acids from the N-terminal side, and then the Y fragments are the ones that are counting the amino acids coming from the C-terminal side. So, for example, here, a B9 fragment would be the fragment or the component that is made up of a peptide that is 9 amino acids starting from the C-terminus, right? And then the Y6 fragment is a fragment that is generated with 6 amino acids coming from the C-terminus. So depending on the sequence, of course, that has a corresponding amino acid weight value, and therefore the component amino acids in each fragment would define the expected mass and therefore also the corresponding charge. And so showing you here are the different B and Y fragments as assigned in the given peptides. Okay. Right. And so depending on that, again, we are able to figure out what component peptides are present. What we try to figure out or through our software, of course, would be what fragments are discernible. And from the discernible fragments, we then try to match that with what we expect to be what we call proteotypic peptides. So proteotypic peptides are the expected fragments that will be generated by a given protease in a peptide, knowing where the protease will cut. So for example, trypsin normally cuts past a lysine residue. So if you have a polypeptide sequence of of a given protein, the potential trypsin cut sites would be where the lysines occur. So given the full sequence of your polypeptide, knowing where lysines occur lets you have an idea of all the potential cut sites. Now, whether or not they would be cut there will depend on the accessibility of the cut site and right, the completeness of the digestion that happens. You know? So depending on the fragments that are generated, one, we are able to identify the proteins that are there, you know, or to verify that the protein is there. Two, we might also be able to identify certain structural features that would have allowed particular cuts or protease cleavages to have happened. So there's actually many types of questions that might be answered depending on how exactly we design the MS experiment and the data that we get from it. Okay, right. Aside from the protein typing that we have, no, sometimes the other important research questions also involve with how modifications can happen, no, in certain proteins. For those of you that are interested in looking at glycoproteins, for example, hindi tayo interesado lang dun sa protein character because we know that some of the functions of these glycoproteins are actually quite dependent on what the sugar residues are that are attached to them. And so that's another area of research, figuring out what types of sugar residues are attached, when, and when these are removed, uh, what changes occur in the function. No? So those are things that we get studied by MS, no? And we can detect conditions, let's say, that might cleave off these particular residues. You know? So if we're testing the possibility that a certain factor can cleave off the residues and therefore provides a certain symptom, we might want to analyze that change in glycosylation through MS. Alternatively, if we want to find out what glycans are there, then you have a glycoprotein perhaps, and then you cleave off the glycans that are there and then are able to analyze the glycan components, then that's another way of analyzing these things. Excuse me. Right. So again, many things to do with the MS. It can even do what we call phenotyping, of course. So this is, for example, looking at different characters that a given bacterium may have, no? Given a certain bacterial genome sequence, what does that mean in terms of its actual character? And that may include, for example, in this case, if you have algal species that provide like high hydrocarbon content, no, this was quite popular in cases where they're trying to make biofuels. Then you might want to see the types of oils that, or lipids that may be generated by a given bacterium. So again, better characterizations of certain sample targets to see if they have relevant importance to us in either the industrial field, the medical field, and the like. Okay, so again, there's many applications of MS. A big part of what we do now is taking genomic data and trying to see how that translates to the proteomic level and the metabolomic level in some cases as well. So aside from the genome assemblies that we have and the genetic data we get, we want to see what is actually expressed. We can do it by mass spec, as shown here on the bottom left. At the PPMF, we actually also do 2D page analysis, which is shown on the top right. No? So aside from MS, where we analyze different protein components through their fragments, we might want to see how the different proteins within the sample actually behave electrophoretically. And therefore, based on differences in the pattern, we might be able to find out if there are distinct changes that occur, again, between two different conditions. So those are things that we try to do. And if you want to know more about that, just ask. Those things are definitely done at the PPMF. Okay. Lastly, again, no, depending on what we have in terms of data, we can look at many different databases. Proteome Discover is, of course, quite useful to us in terms of the analysis. And that's also something that we can try to talk to you guys about. No, if you have data and you're trying to analyze their potential relevance, then we can talk with our team and our consultant team of experts to try to find out how the different ways of analyzing proteomics, metabolomics, and the other multi-omics technologies can actually help in your research question. Okay, so I think that's just enough time to lead up to Dr. Patel's lecture. So I think I will stop here, and I can take any questions that you might have on this very brief reintroduction of mass spectrometry. No? And then we'll go back to Dr. Patel, no? Who will be telling us all the different technological insights on how we can improve our proteomic analysis. So thank you very much, everyone.
Thank you for that wonderful talk, Dr. Vasquez. Our next speaker is Dr. Bhavin Patel. Dr. Bhavin is a senior R&D manager at Thermo Fisher Scientific with a medical degree from the Maharaja Sayajirao University and a master's from Georgetown. Dr. Patel has led proteomics teams at Chase Cancer Center and NextGen Sciences. At Thermo Fisher, he's developed targeted MS assay kits, immunoprecipitation mass spectrometry reagents, and tools for quantitative proteomic assays. Let's all welcome Dr. Bhavin Patel.
Thank you so much for the kind introduction. And can you hear me okay?
Yes, Dr. Patel, we can hear you.
Okay, awesome. I mean, so if you see my screen. Okay, good morning, everyone. I'm really excited to talk about the mass spectrometry solution, consideration for successful sample prep and quantitative proteomics workflow. So today's talk, I will be giving a little background on why protein quantitation is important, what are the different platforms that are used, and what are the two commonly used proteomics, mass spec-based proteomics methods, so discovery proteomics and targeted proteomics methods that are used. And then I'll talk more, the next part of my talk would be talking about the optimized reagents and methods that we have developed to prepare the sample before you do the mass spec analysis. So I'll talk about the EasyPep sample prep kits, as well as talk about the immunoprecipitation to mass spec workflow and PTM enrichment workflow. And the last part of the talk is about getting the quantitative proteomics workflow. For that, we have developed the tandem mass tag isobaric reagents that we use called TMT Pro reagents for the discovery proteomics workflow. And then I will end with the custom peptide that are used for targeted proteomics workflow and how we use all these different reagents for quantitation of the cancer signaling pathways. So in the protein research, there are three main goals. One is to identify the protein of interest, to quantify them and characterize them. In today's talk, I will talk mainly about how much of protein of interest are present in my sample, which means we are quantifying the proteins that are in a sample by mass spectrometry. Now, if you look at the current state of the protein quantitation, this is the current state on the left-hand side. This is the work that was done by one of the graduate students and presented at the AACR Cancer Conference many years ago. And this particular graduate student was studying different phosphorylated proteins that are part of the AKT-mTOR pathway. And they wanted to look at in a 3 different cancer cell line with the different stimulation and inhibitory agents or combination of therapy. As one can see here that you have to run multiple Western blot in order to really assess the differential expression of these phosphorylated proteins across different conditions and different cell lines. What mass spectrometry offers is very extreme sensitivity, and it has inherent specificity. It also quantitates hundreds to thousands of proteins in a single analysis. But not only it looks at quantify the proteins, but it also can identify and quantify isoforms, post-translational modifications like phosphorylation, as well as the protein-protein interactions, and is compatible with the enrichment and isolation methods. So here is the workflow for discovery and targeted quantitative proteomics workflows. So on the left-hand side, in the discovery proteomics, as Dr. Neil was mentioning, you start out with the whole cell proteome. In this case, you can use tissue, cell line, biofluid samples, and you extract all the protein from that sample. And then you do tryptic digestion. Now, tryptic digestion will convert all the proteins and PTMs into tryptic peptide. And this tryptic peptide, you subject them to LCMS analysis by using the shotgun experiment. In this particular experiment, as Dr. Neil was mentioning, you generate the MSMS spectrum to really identify the amino acids that are present for each one of these peptide sequences. And you can do this in a single MS analysis and look at all the different proteins that are identified in particular sample. Now with this discovery proteomics, we are looking at about 1,000 to 10,000 protein targets that can provide relative quantitation of the entire proteome. On the right-hand side is the targeted proteomics workflow. Again, you are starting with similar kind of sample, but instead of looking at whole cell proteome, you already know which protein of interest are present in your complex sample. But you still follow the same sample prep method to generate the tryptic peptide. But in an LCMS experiment, what you're doing is you are actually targeting specific peptides that are part of the unique protein of interest. And what we call that is a targeted experiment. You actually tell instrument to just look for M over Z of those target peptide. Now, when we look at both of these workflow, the sample prep is the similar. It's a common. You follow exactly the same thing for both of these workflow. The difference is actually in your MS acquisition method. So for discovery, you can do label-free quantitation, or you can use the TMT reagents to increase the multiplexing capability. For targeted proteomics, what we do is we develop the targeted assays using the custom internal standard heavy isotope level peptides that you spiked into your sample. And then you can quantify both endogenous and peptide by using the internal standard. So quantitation goals are different between the two methods. When we look at the targeted proteomics method, you can do tens to thousands of targets in a single analysis. What it gives you in a quantitation is more accurate and precise quantitation of proteins of interest. So as I mentioned about the sample prep is similar between the two methods. We have spent a lot of time really standardizing the sample prep methods. So if you look at the workflow on the top, here you are starting with lysis, and then you do reduction alkylation in a sequential manner. A lot of times you are using detergent that are not mass spec compatible. So you have to do protein cleanup by acetone precipitation, and then you do digestion using trypsin and lysis enzymes. And then you have to clean up the peptide before you do the LCMS analysis. Now, traditional workflow takes about 50 steps and 33 hours, so almost 2 days. And some of the challenges with the traditional workflow is you don't get the complete lysis, reduction alkylation, you lose the samples because it's a long process, the quality of the reagents is not good, and it takes a long time to generate the sample. So what we have done is we have developed this EasyPep mass spec sample prep kit workflow, where we actually improve the lysis by removing the sonication step. Here we add nuclease, universal nuclease in a lysis buffer, which actually removes the DNA RNA. And then we combine the reduction alkylation in a one step. And then you do digestion for 1 to 3 hours to clean up in a 0.2 hours. And now you can prepare your samples for LCMS analysis by cutting down the steps to 24 steps and in 2 to 4 hours. So if you look at this EasyPep optimized sample prep workflow, there are two stages of that. So in the first stage, what we are doing is we are chemical and enzymatic sample processing. You can start with cell pellets or tissue that you homogenize into the lysis buffer, or you can start with the plasma and you lyse the protein. Now you're denaturing the protein. And once you get those lyse cell solution or extracted proteins, then you do the reduction alkylation. And in this particular step, because your proteins are denatured, you are breaking up the disulfide bond, and then all the cysteines are protected by alkylating agents, so protein doesn't fold back. And why do we do that? Is because during the digestion steps, you are utilizing the trypsin and lysine. Trypsin cuts on a C-terminal side of lysine and arginine, and that actually denatured protein that doesn't fold back allows the enzyme to actually work more effectively. And in the end, because we are using two enzymes, we can expedite the digestion time to as little as 1 hour. And then you end up with peptides plus contaminant. Now, this is very critical that when you actually get peptides and contaminant, you cannot directly run on a mass spec because it's not compatible for mass spec analysis. Mass specs are very sensitive and they are prone to get affected if you don't have a clean sample. And that's why in a stage 2, what we do is peptide cleanup. Now we bring this peptide, tryptic peptide plus contaminant, and we load them onto these mixed mode resin that are actually packed into this mini spin column format. So when you apply all these peptide contaminants, it's going to bind peptides as well as the contaminant. And then we have optimized wash A and wash B. The wash A will remove hydrophilic contaminant and wash B will remove the hydrophobic contaminant. And then you elute the peptides into a new tube and speed back dry, and then you have a clean peptide that you can use for your LCMS analysis. The reason we went with the mixed mode resin is really to improve the cleanup yield as well as the reproducibility. By doing this, now you can identify, allows you to identify more proteins with 30% fewer manipulation in 60% less time. So as I mentioned about this EasyPep mini spin column format for cleanup, we also expanded the same resin and chemistry into 96 well plate format, as well as the Maxi kit. So with this optimized workflow, you can start out with 1 to 100 microgram of starting material, and you can use the mini as well as the 96 well plate kit. The Maxi kit is designed for starting material of 0.5 to 2 milligram of starting material. And downstream, once you prepare those peptides, you can actually use for label-free quant, or you can do TMT reagents, which I'll talk in a little bit, or PTM enrichment, like high select phosphopeptide enrichment method. The one question we ask, well, if you have different format, how do they perform compared to each other? So in this particular case, we took HeLa cell line, and we actually prepared the samples by using all these 3 different formats. And what you see here is number of protein groups identified by using each format method. So mini, 96 well plate, and Maxi kit allows you to see about 4,500 proteins, 26,000 peptide, and % zero miscleavages are greater than 90%. What % zero miscleavage is that shows how good your digestion is, because there is a possibility that trypsin or lysine is going to miss C-terminal lysine or arginine. So if those peptides are not fully cleaved, then we consider them as greater than 0 miscleavage peptide. So our specs is greater than 90% 0 miscleavages. So the standardized workflow enables efficient sample preparation for 10 to 2,000 microgram of sample amount in less than 3 hours. Now that sample prep method is based on the resin. Last 2 years, we just launched this EasyPep magnetic mass spec sample prep kit. We have 2 different versions of this kit. In this kits, we are using this proteomic magnetic cleanup beads for cleaning up the sample. Now, as I mentioned about traditional workflow, we were doing 50 steps in 2 days. With this EasyPep magnetic workflow, you are still following the stage 1 of the EasyPep method, which means you are doing lysis, reduction, alkylation, digestion together. And then for cleanup, instead of using the resin-based cleanup, you will be using the magnetic beads. Now, the reason you do the magnetic bead method is that you start out with the magnetic bead, you do the binding, wash, and then elute the peptide. This particular method follows very similar like helic-based method, where you are actually doing the peptide binding in high organic and then do the elution in aqueous. With this method, you don't have to speed vector I. Your sample is actually ready to inject in a LCMS analysis. This method even reduces the number of steps. Instead of 24 with the resin method, now you're actually doing only 14 steps in 3 hours. So we have complete kit solution that has pre-formulated reagents and optimized protocol to prepare the protein samples from 15 to 100 microgram with the high yield. And also this method compatible with the TMT and TMT Pro reagents. So what is so special about these magnetic beads? We have looked at so many different magnetic beads that are available for this workflow. And we actually decided to develop our own magnetic bead platform. So this is a new in-house developed magnetic bead platform. These beads are resistant to broader range of pH, like 2 to 11, and it's also compatible with organic solvents and can be automated on a Kingfisher instrument. When you look at the compatibility of this bead with the Kingfisher plastic, our beads are in a green, and these are the steps that you are following to prepare your sample. As you can see, you don't have so much of a bead loss during the bead handling from one plate to the next plate. When we look at our competitors' bead 2, 3, and 4, this bead tends to stick to the plastic. So if you have too much bead loss during the process, you will have more variability and less yield in the end. So this is kind of showing the different kit components and reagents. So on the top part, we have kit to prepare 20 reactions, as well as the kit for 96 reaction. I already talked about the stage 1, which is the EasyPep chemical and enzymatic sample processing. And the step 2 is where you are actually taking the peptide and contaminant and bind them to the beads. Then you wash out all the contaminant, and then you do the elution and remove the bead. There is another workflow that is very popular out there, which just uses the magnetic bead. And in this workflow, instead of cleaning, using the beads to do the cleanup at the peptide level, you can use the same bead to clean up at the protein level. And the workflow for this SP3 method is you start out with the cell pellet, you lyse the cells in a solution so you can extract the protein, you do reduction alkylation, and before you do digestion, you actually bring in the magnetic beads. So these magnetic beads will actually bind all the reduced alkylated protein as well as the contaminant. And then you actually do washes to remove the contaminant. And then you add trypsin enzyme so that trypsin enzyme will actually digest this protein that are already bound to the magnetic beads. And then you separate the magnetic beads, and then your supernatant is going to be the digested peptide that can be run on LCMS analysis. So this SP3 workflow is very short, and it also can be fully automated onto the newer Kingfisher Apex instrument. So how does this magnetic and resin-based method compares with each other? So we actually looked at, in a blue color here is the magnetic beads, and in orange is the EasyPep method. So as you can see here, between the two methods, we are seeing very similar number of protein groups, about 5,400, and about 37,000 peptides. And the yield between the magnetic in a blue and EasyPep method, resin-based method, is about 50%. And we get a really good digestion efficiency up to 95% zero miscleavages. So the both chemistries and both methods are very identical. It's really users' preference in whether they would like to do automation on a Kingfisher, then they can automate this magnetic bead workflow. When we look at the overlap between the replicates using this magnetic bead method, at the protein group level, we see between the 3 replicates, we see almost 94.5% of the peptides that are common. And at almost 90% of the peptide that are common and the 95% of the proteins that are common. So it's very reproducible. And this workflow is also compatible with TMT. And I will talk a little bit about TMT and how TMT works and also improves your multiplexing capability. So as I mentioned about the SP3 method, where you actually don't have to use the resin, or you can do the protein level precipitation and cleanup. We took the HeLa cell lysate 100 microgram, and then we prepare in a multiple replicates. In this particular case, 3 replicates. And we were able to see almost 6,000 proteins and 66,000 peptides from this sample prep. When we look at the overlap between the protein groups, we get almost 99.4% overlap for the protein. And we look at the median CV, we are getting 4.4% median CV between the replicates. So this sample prep method using the magnetic bead is really reproducible, and it actually provides excellent quantitation reproducibility. One question a lot of people are asked, like, what's the range of the starting material you can use and what different types of sample can be prepared using the magnetic bead workflow? So in this example here, we looked at 1 to 15 microgram, 15 to 100, and 150 to 1500 microgram. And you can see we are from the HeLa cell line, we are getting consistently 5,900 proteins and about 62,000 peptides. So it's really reproducible. And we also looked at different sample type. So besides cell line, can we use plasma sample? So if you have plasma that you digest, you get about 300 proteins and about 3,300 peptides. Now this number is very low, and I'll talk a little bit about how to improve the proteome depth in a plasma samples by using some depletion reagent. When we look at the tissue and E. coli, we are seeing in a tissue about 3,200 proteins, 30,000 peptides, and 2,200 proteins in E. coli and 25,000 peptide. If you look at the error bar, the CVs are really low. We see consistent ID within 5% CV between replicate across all the sample range and sample type. So this proteomics magnetic cleanup bead sample prep are compatible with wide variety of sample types as well. So this is our general sample prep methods that we have optimized. One is resin-based and one is magnetic bead-based. But when we think about mass spec sample prep workflow, it's really based on the protein abundance. So let's say if you are only interested in high abundant protein, you can use this MS sample prep method and can see the top 6,000 proteins in a cell line. But when we think about plasma or some other biomarkers that are really, really low abundant, and a lot of times when you look at the protein abundance, it's actually 10 to 12 orders of magnitude dynamic range. So a lot of the low abundant biomarkers are not really highly expressed. So when you look at the 6,000 protein ID in a cell line or tissues, you're not going to be able to see those low abundant targets. So there are a lot of different strategies that we use to improve to address the dynamic range and allow detection and quantitation of medium to low abundant target. One strategy would be to deplete the abundant proteins. Another strategy would be to do enrichment. Maybe this is a target-specific enrichment before you do the digestion. So the first method that we have developed is depletion method, and this is specifically applies to human plasma serum and CSF sample. The more common method of depleting these high abundant proteins, such as albumin and IgG, which makes up greater than 80% of the proteins in a human plasma, and that really hinders the detection and quantitation of biomarkers. There are column-based methods that are available. The issue with that is actually introduces more variation, carryover, and contamination because you're using the same column multiple times. So what we have done is we developed this spin column format, and that has a resin that has antibody that are immobilized onto those resin. So this spin column method is fast, reproducible, and very cost effective. And they are only single use. We have two different formats of this, mini and midi, that process up to 100 microliter of plasma and serum. And you can also get the resin by themselves. So what are the abundant proteins that we deplete using this column-based method? So there is top 2, which depletes the human serum albumin and IgG, and all different types of IgGs. And then the top 14 columns depletes additional protein that are actually part of the top 14 abundant proteins. If we look at the gel here, it actually shows how efficiently we are removing some of this abundant albumin and IgG when you look at the flow through. So what you're doing here is you incubate your plasma with the resin, and all the abundant protein will bind to the antibody, and your flow through is your depleted sample. In the top 14, you are depleting even more. So now you start seeing very low abundant proteins that are present in your flow through sample. We also looked at the depletion efficiency using ELISA kits for each one of these target, as well as the mass spec analysis. And we see very good, almost greater than 95% depletion efficiency, and for most of them is 99 to 100%. So how this depletion resin compares with the other competitors' product. So high select top 2 and high select of 14, you load about 10 to 20 microliter. Agilent top 2, Agilent top 14, and Sigma top 14. They are using about the same amount of sample load. The difference is in a buffer volume. So we have optimized the buffer volume and reduce it. So now you have only 100 or 200 microliter of buffer compared to 400 to 600 for other competitors' product. And why this is necessary? Because when you deplete, you will end up with a more diluted sample with the competitor's product. With our product, you are going to have only 100 to 200 microliter of depleted sample, which allows you to not do the concentration. Because whenever you do concentration or concentrate the sample, you're going to introduce more variability and loss. So how do we put the depletion resin with the EasyPep kit and generate, ask a question, what are the differential proteins that are present in a normal and cancer plasmas? So in this particular example application, we took 4 different normal plasma from 4 different normal people and the plasma samples from 4 different lung cancer patients. And then we actually did the depletion in a 96 well plate format. So this well plate format, you can dispense the resin into each individual well, and then you add the plasma that are from each one of these different samples. And then your depleted sample, you can perform the EasyPep 96 sample prep method to generate the clean peptide. And then it is critical to quantify this peptide before you do LCMS analysis. So we have developed these peptide assay kits. This is very similar like how you do protein assays using BCA reagents. This one is actually for quantifying peptides. And because you can quantify the peptide, you can normalize the load on when you do the LCMS analysis, and then you do your data analysis using the Proteome Discoverer software. So in this particular plasma proteomics application, comparing normal and cancer, the left-hand side is the PCA plot. Here you can see that we are getting 1,500 unique proteins. So when we don't deplete, we are seeing only 300 proteins, but now we can see 1,500 unique proteins and 10,000 unique peptides. And each one of these patient samples, replicates, are grouped very well. And when we ask question about some specific biomarkers, their expression, here one of the example of the lung cancer biomarker, mucin 5B, is specifically increased threefold into the cancer patients compared to the normal and other cancer types. There is also another cardiovascular biomarker, cadherin 5, that increases twofold in a cancer versus the normal patients. And there is another oncology biomarker, CD44, showed 1.5-fold increase in a specific patients compared to other samples. So this plasma proteomics using the top 14 depletion column and EasyPep sample prep kit allowed the quantitation of these disease-specific biomarkers. Now, another method that a lot of times researchers are looking for is enriching the very low abundant targets. And this is very similar like how you do the immunoprecipitation. In this particular enrichment method, you incubate your antibody, target-specific antibody with your lysate. You, from the immune complex, you do the washes to remove the detergents, and then you do the enzymatic digestion or elution, followed by another digestion, and then you run the samples on LCMS analysis. So this optimized workflow with using the enzyme to do elution and then EasyPep method to prepare the sample. And this takes about 4 hours. So we have developed these 2 different kits for IP mass spec workflow. They both uses the magnetic beads. The magnetic beads has protein AG or streptavidin immobilized onto them. And the both kit has all the reagents that are necessary to prepare the sample for mass spec analysis. So here is an example of using this IP mass spec kit to enrich multiple biomarkers from the human tissue samples. So if you look at on the left-hand side, we took multiple targets from AKT-mTOR pathway. If you remember, I showed you the Western blot with many, many Western blot that you need to run to assess the expression between the sample. In this particular case, you are doing immunoenrichment by combining antibody that are specific to each one of these target in a single IP using the IP mass spec kits. And then you actually do the quantitation by spiking in heavy peptides for each one of these target. And you can see we were able to quantify all of these different AKT-mTOR pathway biomarkers between the breast normal and breast tumor sample. So you can see here, breast tumor actually expresses the higher abundance of these biomarkers. When we look at the phosphorylated forms of the same AKT-mTOR pathway targets, we use the antibody that are specific to this phosphorylated form of target, and we were able to multiplex IP them and do the peptide quantitation to look at the differential expression of these targets. And you can see here in a tumor, some of these pathway phosphorylated targets are very highly expressed. So it actually confirms that when you have a cancer, this particular pathway is activated. So the next part of the talk, I like to discuss about the multiplex capabilities can be done by using this tandem mass tag. So here on the left-hand side, we have developed these TMT Pro 18-plex reagents. These isobaric tags are very unique in a way that the total mass of this tag is similar across all these 18 different reagents. The difference is actually in the reporter group. So the way it works, that these are amine reactive tags. What you do here is you actually take 18 different samples with the different conditions, and then you do the EasyPep sample prep method to generate the peptide. And then you label each one of these digested peptide sample with individual tag. Then you combine them together, and then you fractionate, clean up, and do LCMS analysis. Because these are isobaric tag, when you do MS1, they all are stacked up as a single M over Z for each peptide. But when you do MSMS fragmentation, the reporter ion is a different mass for each one of these tag, and that will be resolved in your MSMS spectrum. So here you can see this is a resolved spectrum for all these 18 different reporter ions. And by doing this way, you can actually not only identify the specific biomarker, but also you can quantify in a multiplex format, which means you can combine 18 different samples in a single analysis. How does the TMT reagent fits with the EasyPep workflow? So if you remember, we start with the stage 1 and we get the peptide plus contaminant. You can actually label at this particular step, or you can actually do the cleanup and label the clean peptides with the TMT reagents. Both these approaches are viable. There is no preference one versus other. It's really up to user. If you are doing the labeling after cleanup, you still have to, with both these methods, you still have to fractionate the sample in order to get much deeper coverage for your proteome. Because these tags are previously provided in a dry powder format, we more recently actually developed the solution stabilized TMT and TMT Pro reagents in a 96 PCR plate format. So if you see here, we have actually different variants of TMT reactions for doing 10-plex, 11-plex, or 18-plex, or 16-plex. And these are pre-formulated, each one of these tag in an individual well. So customer can buy and use whatever the number of wells they want to use for their multiplex experiment. So in this particular example, we did TMT labeling for HeLa digest samples. They are from 1, 5, or 10 microgram. And we can see about 4,000 proteins, 25,000 peptides, and the labeling efficiency of the TMT is greater than 96%. For most of the 5 and 10 microgram, you get 99% peptide label with the TMT. So these are the 18-plex tag. More recently, we actually expanded the 18-plex to 32-plex. So now you can actually multiplex 32 samples in a single analysis. And there are different variants of this tag. The question is how will we be able to make 16 to 32-plex? The way we did it is our 16-plex tags, we actually utilize the deuterium as a defect to generate the additional 16. So each one of these TMT Pro reagents, we have a matched deuterated tag for as another tag. So now by combining 16 plus 16s, you can actually do 32-plex. The question is when you do the higher multiplexing, how does it really helps to reduce the complexity? We looked at the 18-plex and 18-plex doing twice versus 32-plex. And if you look at the number of quantified proteins, you do get slightly lower number when you do 2 different 18-plex separately. But if you combine 32-plex, you get plus 15% higher number of protein and 39% increase in a peptide identification. And one of the key thing with this quantitative proteomics is the missing value. If you run the same sample multiple times, you will start seeing very overlap of the protein and then unique protein that are in one replicate but not in the other replicate. And that's the problem with the quantitative proteomics. So this TMT allows you to address the missing value problem, and you can now see all the proteins that are actually present in a single experiment. So here is an application of how we integrate the sample prep and TMT to look at some clinical samples. So these are FAPE samples. We start with 6 different tissues, normal and cancer, and then we extract the protein from this tissue. So we have a protocol that is optimized to extract the protein from FAPE sample. And then you do EasyPep to generate the peptide. You do TMT Pro 18-plex labeling, then you combine, and then you do quantification. So that way you know that each one of these tag is labeling exactly same amount of peptide for each one of these 18 samples. And then we do fractionation, LCMS analysis on a tribrid instrument. Now this tags does require high resolving power, so they are more suitable for running on a tribrid instruments. And then you do the data analysis using the Proteome Discoverer software. So here is the PCA analysis of the results. So these FAPE samples, we were able to, you can see they are nicely clusters for colon tumor and normal, lung normal and tumor, breast normal and breast tumor. And in terms of identifying proteins and peptides, we were able to see 4,000 protein and 26,000 peptide with the TMT Pro labeling efficiency of greater than 98%. So it really provides a very powerful tool to look at the biomarkers from between normal and cancer samples. And this is specifically for FAPE samples. On the right-hand side is an example of some known breast cancer biomarkers like ERBB2. You can see 6-fold increase in a tumor breast tumor over normal. And then colon biomarker CA-CEACAM5 was 2 times highly expressed in a tumor versus normal. And then the lung cancer biomarker coronin 1A is 2.5 times highly expressed in a lung tumor over normal samples. So it really allows you to do the protein biomarker analysis between the normal and disease samples. So in the last few minutes, I'll talk about targeted method. So um, when you think about targeted proteomics, you really have to start out with understanding the sample prep method. You have to select the peptides that you can actually synthesize them in a heavy isotope label form. This peptide has a heavy lysine or arginine on a C-terminal side, which allows you to spike into your endogenous samples and quantify the amount of endogenous peptide by taking the ratio of light or heavy. And then on an instrumentation side, you have to develop the targeted method so that you can ask instrument to just look for the light and heavy pair of those M over Zs for each one of these unique peptide. And then you do the targeted assay MS acquisition using the LCMS system. And the data analysis is done using more commonly used using the Skyline software. So here is an example of the 30 unique peptide that covers the 12 AKT-mTOR pathway targets. So we have developed this. We use the aqua heavy peptide. These are ultimate grade peptide that has high purity greater than 97% and concentration precision of less than 5%, and then isotopic enrichment of 99%. As you can see here, each one of these peptide has a very broad dynamic range, about 2 to 3 orders of magnitude, with the lower limit of quantitation as low as 0.5 femtomoles on a column. And when you look at all these different peptides, most of the peptide we were able to see with the very lower limit of quantitation. So how do we use these peptides to do the targeted signaling pathway analysis? And as you can see here, there are many targets and many different signaling pathways that crosstalk with each other. So one way to do it is you can actually enrich multiple targets, as I talked about using the IP mass spec kits. So in this case, we took cell line and tissue, and we enrich all the total and phosphorylated targets using the multiplex IP, and that can be automated onto Kingfisher platform. And then you do spike in heavy peptides and aqua-grade peptides to do the quantitation. We applied this IP mass spec and heavy peptide quantitation methods for the lung cancer xenograft samples. So this is the work that was done with Dr. Mike Moran's lab at the Sick Kids Hospital in Canada. And they were able to actually quantify all the 12 different phosphorylated forms of the AKT-mTOR pathway targets across all different lung cancer xenograft samples. So this method is very powerful to get accurate quantitation. When we look at the analysis of multiple cancer signaling pathways, so instead of just looking at AKT-mTOR pathway, you can actually look at 7 different more heavily studied cancer signaling pathway, as shown up here. What you're doing here is you start with your sample and prepare the digest using EasyPep method. And then you'd have the tryptic peptides where you can spike in this aqua grade heavy isotope level peptide to your endogenous peptide. And then you do the phosphopeptide enrichment using the high select reagents. The reason you have to do enrichment for phosphopeptide because the phosphopeptides are less than 1% of the total peptides. So when you just run that sample as is using mass spec, you're not going to be able to see a lot of those phosphopeptides. So we have developed the magnetic bead-based INTA enrichment method to enrich the phosphopeptides from the complex tryptic peptide mixture. Now you're enriching both endogenous and the heavy isotope level peptide, and then you can do the targeted experiments to quantify each one of these peptide. So we shared these reagents with one of our collaborator, Professor Jesper Olsen at the University of Copenhagen. And they actually had a control cell line, and then they stimulated with EGF for 10 and 22 hours, and IGF for 10 minutes and 2 hours. And as you can see here, this is a quantification of 103 phosphosites that covers all the 7 different cancer signaling pathways. And now by doing this targeted method, you can really see the changes in the phosphorylation status for each one of these phosphoprotein in a single analysis. So in a summary, really the mass spec sample prep method, including the IP mass spec kits, really enables the single-day sample prep method. Reproducibility and efficient sample preparation is really key for all the successful proteomics experiment. And with the EasyPep sample prep method with the resin or magnetic bead based, it really provides the effective solution to prepare the high quality proteomic samples for LCMS analysis. And enrichment using IP mass spec kits really allows you to do the quantitation of low abundance signaling pathway proteins as well as the PTMs. And the TMT, it really takes it to the next level. You can really multiplex your multiple samples in a single analysis that provides the high accurate and precise quantitation. So with that, I'd like to acknowledge a lot of my colleagues at the Thermo Fisher, collaborator at the Sick Kids Hospital, University of Copenhagen, Proteome Sciences, as well as the Harvard Medical School. Thank you so much for your attention and happy to answer any questions you have.
Thank you, Dr. Patel, for that powerful presentation on sample preparation for quantitative proteomics and its application on health research. So now we'll open the floor for questions for Dr. Patel and Dr. Vasquez. So please type in your questions in the question and answer box.
Yeah.
Okay, while waiting for the questions, I would like to ask, Sir Neil, can you turn on your video?
Hi, Victor. My video is on. Can you see?
Yes, yes. Okay. So while waiting for questions for Dr. Patel's presentations, there's a question here for Dr. Neil on your previous presentations. What samples do you use in proteomics, blood or tissue samples, and what sample preps do you usually use?
Okay. On what samples we use in proteomics, it depends on the research question again. For example, we've had a chance to use plasma in some cases, and in some cases we've handled urine. So it would really depend on what tissues might be relevant for that particular research question. For example, again, kung diabetes, then one obvious thing would be to look at the urine because of potential kidney function problems. In terms of sample preparations, we've tried several methods, and definitely we've had a chance to test as well, no, the available kits from Medtest, and they've been quite useful.
Thank you for your answer, Dr. Neil. So the next question is for Dr. Patel. This is from Mr. Ed Montera. Thank you for your comprehensive and wonderful discussion. As a beginner in mass spectrometry, what are the common mistakes a researcher makes in sample preparation and how to avoid them?
That's a really good question. I think if you look at back the traditional workflow that people use, and really they try to use different reagents, they follow the protocol that's available on a published in a research articles. The critical thing with the mass spec sample prep is really understanding what are the right reagents that you are using to denature the protein. Are you getting good reaction alkylation? That actually ties with your digestion efficiency, because anything you do upstream that doesn't have a good lysis, you don't reduce alkylate very well, you use poor quality reagents or not optimized conditions, it is going to affect your digestion efficiency. When you have the incomplete digested peptide, it's actually going to create more variability for the same sample you prepare multiple times. So that's very critical to really understand the type of sample that you're using and what is the right sample prep method to use to extract the protein, reduce alkylate them, and get the near complete digestion for before you do the cleanup. Cleanup is also very critical because a lot of times when you are new, you don't know what detergent can be removed with what resin chemistry. So that's why a lot of times somebody say, okay, I'm using SDS and I can use C18 to do the cleanup. It really doesn't work that way. SDS will bind very effectively to the C18 and it will come when you elute your peptide, and now your mass spec is going to be contaminated, as well as your data is not going to be great because SDS suppresses your signal. So it's very important to really look at more standardized workflow solution. Our approach is, our efforts has been to really develop the standardized kits specifically for people who are new to the, proteomics and mass spec, where they can actually use those reagents and generate the high quality sample. So really understanding like your sample type, let's say if you are using tissue, you need to make sure that you follow the protocol to homogenize the sample so you get the maximum amount of protein out. If you are using plasma, you need to also make sure the quality of the plasma is good and also handle them as using the proper steps, and then use the standardized kits to follow to prepare the sample. It's very important to have consistent extraction, digestion, yield by doing the cleanup so that you can actually trust the peptide and protein that you're identifying in a mass spec analysis.
Thank you, Dr. Patel, for that answer. So our next question is from an anonymous attendee. So the attendee is a marine biologist currently working on a DNA barcoding of ectoparasites of several marine fish species. So he was asked, or he was asking if he would be able to apply proteomics in his study. For proteomic analysis seems like a good confirmatory or an advanced application of my DNA barcoding approach.
So if you are using like samples, as long as you can extract the protein from those samples, you would be able to apply the proteomics on them. Now, if you, for DNA, sample prep is different than doing the protein sample preparation. There are some efforts and there are some articles out there how to extract the different analytes from the same sample type. So I would say that you can extract the protein from these samples and do the sample prep using the EasyPrep method that I talked about, and you can actually run them onto the mass spec instruments to generate the proteomics data.
Thank you for
Victor,
that
I think,
answer.
sorry, if
Sige.
I may add to that one. So given that the current study is more on DNA barcoding, it would depend on what features of the genome are actually available for the sample. Because sometimes when we do barcoding, right, we choose specific, easily targetable barcoding genes to be able to distinguish the different species. But whether or not the differences in the barcodes translate to particular characteristics of the species that are easily distinguishable, that would depend on what other genes are present in them. So I think definitely an expansion of that study could be either full genome analysis of what is compared, and then from there, translation into what are the likely proteins that are going to be um, differentially present between the two different species. So that's one thing to look at, and definitely multi-omics analysis can be helpful there. Sorry, Dr. Balvin, just as a follow-up also into your question, your answer on this on whether or not you could have multiple samples, similar samples analyzed with different omics methods. So you're mentioning how the ECPEP system can probably try to extract from samples that have, previously been processed for DNA extraction. Is that correct?
So DNA with the EasyPep method, what we have done so far is really just remove the DNA RNA. So not really can be used as a multiple analyte method. We are working on a newer program, hoping to
Okay.
launch early next year. That actually allows you to extract DNA, RNA, and protein sequentially. So you get the protein out, you have the DNA, RNA left, and then you actually use the remaining samples to separate the DNA and RNA. And this is going to be a magnetic bead-based method to
Right.
do the multi-analyte analysis. So kind of extension from our EasyPep magnetic workflow, but it's using a different beads with the different properties that allows you to separate the protein, DNA, and RNA from the same sample.
Right. That sounds great. I think especially as people are moving into more multi-omic analyses, when you can extract the same type of material that you will analyze for the genome, transcriptome, proteome, and metabolome all from the same sample, I think it's really much better in terms of linking them together. Yeah.
Yep. Yeah. I think that we are very close to kind of finishing up the validation, and we should be launching that product early next year. Yeah.
Great. Thank you.
Thank you for those answers. So next question is from Miss Aquino. So it's a very general question. What are the limitations of MS analysis?
Who's taking it? Dr. Balvin, you want to go through this?
Yeah. So I think the limitations of mass spec analysis, I would say that the mass specs are getting more sensitive than 10 years ago, specifically last 2 years with the new instruments that are developed and analyzers that are developed. Now you can actually go up to like 10,000 proteins from a given sample. They are getting more faster, more sensitive, and with the more label-free quantitation, you can actually go deeper into the proteome. But having said that, you're not going to be able to see, um, all the proteins that are present. So when you think about other immuno-based methods like ELISA or Western blot or Luminex, they are more sensitive. So that's one of the limitations that we are making progress in terms of instrument sensitivity. We are made some good progress on enriching the phosphopeptides, but some of the other PTMs can be a lot more challenging as well. So that's one of the limitations, that sometimes biologists who's interested in a specific protein, if it's very low abundant and you do the mass spec analysis, you may not see that particular protein in your data. So that's a challenge. Another limitation would be adoption, wide adoptions or ease of use. I think these are more specialized techniques and requires expensive instrumentation and trained personnel to really execute a successful mass spec experiments. So that's kind of, we are making progress to make it ease of use, more hands-free, more standardized reagents, kits, but still it's not at the same level as when you think about like next-gen sequencing or some of the other methods like Western blot or ELISA. Where you advantage, I would say, that mass spec provides compared to other methods is specificity, because you are actually confirming the peptide sequence or PTM confirmation. I think the other methods are relying more on a like orthogonal approach to say, yeah, this antibody binds to this target, but they also binds to 10 other things as well, or could bind. So that specificity is the big advantage for the mass spec methods. Dr. Neil, feel free to add more things.
All right. Thank you, Dr. Patel. So I think I'll just add a little bit.
You are muted.
Okay. So definitely the specificity that mass spec provides is one of the key things, no? And um, it is a gold standard in trying to confirm, certain the presence of certain things. Now, in terms of limitations, as mentioned before, in the way that we analyze, because this is a very sensitive system, um, it's quite dependent on how well you process the samples that you put in, no? And so and part of really ensuring that you get great data comes from that initial sample processing, no, that needs to be well defined and done correctly by trained staff, and et cetera, et cetera. So it was also mentioned that in terms of adoption, it's quite hard sometimes because the facilities or the infrastructure might not be available. In that sense, I think there's good news that there is great growth in terms of that. There's expansion of capabilities that is being provided in the Philippines. And I think the more people get used to doing the work, the more proficient we all become, no, in being able to process the data, get the data properly, and analyze the data to make it more applicable to our own applications. And so that's one thing. Another is, of course, it's a destructive method. So sample availability will be an issue. So if it is very minute, and like Dr. Babin was saying, if there's small amounts of the thing that you want to analyze, um, well, how it should be handled so that we are able to accurately detect that target. No? So it all boils down to good research design. No? So I think that's what we need to do.
Yeah.
Mhm.
And if I can add just one more thing, right? I think the key part is understanding your objective and utilizing the more standardized sample preparation method um, to really get high quality mass spec data. I would say that, last 5 years, there is a significant growth in instruments capabilities, more robust LCs, more standardized sample prep methods. It's really allowing us to actually look at the biology that we were not able to do by mass spec before. Um, and I would say that, there is people always think about mass spec and data as like, quite overwhelming, but I think we are making a huge progress on getting the data analysis and visualization even more easy for biologists to understand. So
Thank you for those answers. So for the next question, we have a question from a biologist, and the biologist wanted to integrate the liquid chromatography mass spectrometry for the quantification of specific compounds from microorganisms as part of his study, and he really wanted to integrate proteomics with this. So, are there any insights or suggestions on how to do so?
Victor, can you repeat that?
Okay, okay.
Nag-copy lang. Okay.
Hi. I'm a biologist and I wanted to integrate LCMS for the quantification of specific compounds from microorganisms. So I really wanted to integrate proteomics with this. Are there any insights or suggestions on how to do so?
Okay. Dr. Patel, if I may. Very, very
yeah,
briefly.
please.
So if we're looking at the compounds that are being produced by the microorganism, I think the first approach should be a metabolomics approach, because that would be more directed towards these specific products. So I think that's definitely one way to do it. Alternatively, if we want to confirm if the necessary proteins for the production of these products are actually there or are affected in a particular way given two different conditions, then you may follow up a metabolomics level analysis with a proteomic level analysis, wherein you now target the expected relevant proteins for the expected products as your, as your analytes, no? So as you what you might want to quantify in the different conditions. I think that's two approaches that we may be able to do. And both should be available at PPMF. Dr. Patel, maybe additional.
No, I think you covered it. I think the metabolomics is the way to look at the small molecules, and the sample preparation is very straightforward with that method to really look at what those compounds are.
Okay, thank you. And next question for Dr. Patel. When using a plasma or serum sample, how can we address the low signal in MS?
So when we think about using plasma or serum, I think I talked a little bit about the dynamic range issue, right? So when you have such a high dynamic range of 12 orders of magnitude, where 80% of the abundance is coming from albumin and IgGs, you're not going to be able to see low abundant targets. So in order to address that, there are a couple of strategies that are coming more recently. One is the depletion method that I talked about, where you deplete the more abundant proteins that allows you to enrich the low abundant target. There is another method, what we call is protein corona isolation. So it's an enrichment method. In this particular case, you are using the magnetic beads to capture all the proteins, and then you remove some of the abundant proteins. And there is a different way to do that. There is a paper that came out a couple of years ago that uses the magnetic beads called magnet method. In this particular case, you are using the strong anion exchange beads to capture this protein corona from the plasma and serum sample. The challenge with that approach is you have to use the unfiltered plasma. You cannot use the filtered plasma. So it's a very limited approach in order to address this dynamic range issue and reach the low abundant proteins for mass spec analysis. There is another approach called nanoparticles. That's something that I didn't talk about today. And in a talk, we don't have a current solution using the nanoparticles to enrich the low abundant proteins, but there is some of those solutions are out there. And that actually allows you to now go up to 4,000 to 5,000 proteins from the plasma sample. So it's really, there are some new sample prep methods that actually allows you to get low abundant plasma protein detection and quantitation in a mass spec.
Thank you for that answer, Dr. Patel. So another question from Mr. Montera. So he has two questions. Are there any ways to verify the sequence of the protein after performing MS? And second question, is the workflow in MS during de novo sequencing any different?
I guess I'll take that one. After identifying the protein, the protein target, that the way that we actually identify it is because the fragments that were identified within the sample match expected mass to charge ratio values that should be, come together to put up the source protein. And as part of our QC control, we actually look at how much of the expected fragments are actually observable in the sample. So in a way, those that can be reported with high confidence are the ones wherein all the expected fragments, let's say, are actually there, no? And in already that should provide a sense of what the protein sequence is for that particular target. Alternatively, though, should you choose to analyze, let's say, a single protein target, that is a possibility. If you are able to extract that single protein target and then fragment it let's say, by trypsinization, that then comes up to expected fragment sizes and mass to charge ratio values, which you can then verify through mass spec. So that's one other possibility of trying to check if what was identified before matches, let's say, a recombinant form of this protein that you would want to check is present also in the sample. I hope that makes sense.
Mhm. I thank you, Dr. Vasquez, and I can take one last question from Mr. Martizano. What is the minimum mass analyzer resolution required for proteomics, and which type of mass analyzer can achieve this resolution?
Dr. Patel, maybe you'd be a better resource for this question. Yeah.
Yeah. So I think the when you think about mass analyzers, uh, I think there are two more commonly used mass analyzers. I would say three, right? You have TOF analyzers or Orbitrap analyzers, or more recently, we have commercialized 2 years ago, or it's called Astral analyzers. Now, the resolving power of these analyzers are different. The Orbitrap is the only analyzer that actually has a resolving power up to fourth decimal points. So when I talked about TMT, isobaric tag, these tag mass are so uh, close to each other, which means they are only differ at the second or third decimal point. You need to have very high resolving Orbitrap mass analyzer in order to separate those two reporter ions from each other. So if you are looking at multiplex discovery proteomics experiment using isobaric tag, you really need to have mass analyzers that has a resolving power of like 240K or 480K, or even higher than that. Higher the multiplexing for TMT, you need a higher resolving power Orbitrap analyzer. Now, when we look at the other QTOF or like TOF or Astral analyzers, their resolving power for doing the MSMS and just identifying the peptide sequences, you can be okay with having like, 60, 90 K resolving power. You don't really need a very high resolving power because you would be able to actually get a good MSMS spectra with that low resolving power. The advantage with those analyzers is faster, and you can actually get more data point across the peak. So it's really depends on whether you are more interested in a discovery proteomics with label-free approach versus discovery proteomics using the isobaric tag like TMT Pro reagents.
Thank you for that answer, Dr. Patel. But I would just like to ask one very last question. So we usually do nanoflow for our LCMS proteomics analysis here. So my question is, can we do a microflow for proteomics, and what are the drawbacks as compared to the nanoflow?
I would say nanoflow is really allows you to um, be more sensitive, right? So when you think about microflow system, you are looking at anywhere, um, you know, first of all, you will have to load more samples onto the column. So you definitely get a better throughput, uh, better like, uh, reproducibility, I would say as well. But it comes with the expense of lower sensitivity. And you will have to load more on a column, and that way you will be able to, get a better resolving power. The nanoflow is actually more heavily used on a discovery proteomics side, where you're really trying to get more sensitivity, and you don't need to load so much onto the column. You can get away with like 100, 500 nanogram, or 1 microgram is what typically a lot of people load onto the column. The challenge with nanoflow is that you have to run a longer gradient, and you will have to your throughput actually gets you compromise throughput over sensitivity, right? So that's the trade-off. So whenever you are designing an experiment and the objective is to go deeper proteome, nanoflow method is a better way to do it. Let's say once you identify those biomarkers and you want to run many samples for few targets, microflow would be a better way to do it. Now I would say nanoflow with the more newer system, you can actually run 30-minute gradient or even 15-minute gradient. So a good balance would be nano to capillary flow. So like if you do 300 nanoliter per minute or 1 microliter per minute, it's actually a better compromise than going like 10, 40 microliter per minute.
Thank you for that answer, Dr. Patel. So this officially ends our question and answer portion. So if you have any more questions, feel free to type it in our question and answer box. We will send it to our resource speakers. And I would just like to inform all of you that Dr. Aaron James Reyes has planned to speak today, but he's had a medical emergency. But no worries, after this webinar, we will just send his lecture to all the participants. And this concludes our program. And now, to formally thank our speakers, we'll now present the certificates of appreciation.